And also you can contact me if you have any questions on how to do this. Okay, so this is one of the herbarium labels. Amber, who used to work at the herbarium, now she's working at Kew. She wrote this and sent this to us. So this kind of breaks down exactly what goes onto a specimen label. So instead of flora, ours will say fungi. So this is representing a plant specimen. And then it has the scientific name and then the genus species name. And then it goes into more detail with locality of where the sample was collected, which is in purple, and then habitat. So where it was found, what type of forest was there. And then it goes into any characteristics of your specimen. So what color it was, that's probably one of the most important things. If it is any particular color, that's going to be lost as it dries. So you do want to put any information about color, about whether it has an annulus or a ring around it, especially if it's ephemeral. If you find a puffball, those usually stand up easier to drying. And then after that, you have the coordinates and elevation, and then the date it was collected, and then who the collector was. Oop. Okay, so this is kind of the most detailed label. Yours does not have to be this detailed, okay? This is a lot of information. Like if you do make a label like this, that's awesome. It has a lot of information, but it does not need to be that detailed. And I will give an example of a slightly less detailed one. So this one is one that we collected that we're submitting tomorrow. So I have, again, fungi of Texas, and then the family and genus and species. And I do want to point out, I talked to Amber and George, who work at the herbarium, about this. You don't have to have it ID'd down to species if you have it as you just have the genus, or you just have the family name, or you just have that it is Basidiomycota. That's completely fine. They will accept anything, and someone else will identify it for you. Okay, and so then again, we have location in purple. So this was collected at Stengel Lost Pines Biological Station. Any information, like you want to think about how would someone find it if they were going to go out there and look at this mushroom, exactly where you found it. You want to be able to describe that to someone. And so you don't want to put anything that may change. So if there is a house that's red, if you think like 100 or 200 years from now, that house will likely not be there, or it will at least have changed color. So you want to do something that will be there many years from now. So here, there was a trail. This is not very detailed. This is not the best example. But at Stengel, they have a grid system. And so ideally, we would put where on the grid system it was. In blue, we have the habitat, so what type of forest we were in, oak, juniper, pine forest. And this was collected from the soil. So substrate is also something that's really important. Was it collected from a tree? Was it found, if you're doing a lichen, was it found on a rock? Was it found on soil? And then here we have some information about the morphology and the growing habitat of it. So it was growing in a group. As you can see here, it's these really tight groupings, which is kind of indicative of this species. The color was yellow, dark cap, and then also a partial veil or annulus. And I have the specimen here actually, and you can look at it and see how much it's changed from this. Okay, so let me actually, I'm gonna run over and grab those specimens.
Okay. Questions that came up in the chat too that we can go over before we get into looking at these specimens.
Sure.
So I'll hold on and I can ask it on the mic.
Oh, yes.
That way everybody online. So he asked, can the short-term storage in non-archival packages in order to carry the sample to the herbarium be an issue? So how do we determine that a sample is appropriate for inclusion, not a duplication? So there are two questions there.
Those are very good questions. So your first question was you were worried about whether what you put it in may compromise the quality of it. In general, if you just make sure you use like a clean Ziploc bag, it should be fine. Some of them do have like a powdered residue that they put in it to prevent it from sticking and to keep it dry. But as long as it's, it's not a ton of that in it, like if you can't see it with your eye, it should be fine. If someone wants to use it for DNA, they can just cut it open and take a sample from inside the mushroom. So I think that should be fine. And then your other question was about, can you remind me?
How do we determine it's not a duplicate?
Oh, duplicates. So duplicates aren't a bad thing. Actually, that's good. So if you actually right out here, I've collected the same mushroom, I think 3 or 4 times now. And duplicates aren't bad because you can follow it through time. You can see what seasons it's coming out. And then, you can follow it over the years. Like it was found here 10 years ago. And then 5 years later, it was found again. 3 years later, it was found again, and it hasn't been found since. So is it gone from that area, or is it just the environmental conditions aren't right? So duplicates are completely fine. And I encourage you to do duplicates. If they have too many of one type from the exact same location, George at the herbarium may say, okay, I don't think we want this many, but in general, I think it will be completely acceptable. Good questions.
All right, so we can stop sharing the screen and then I can switch to.
Yeah, thank you. Okay.
All right, everyone. So now we are.
Okay, so you guys can see this up here. And I will pass these around too so everyone can see them.
Yeah, actually.
Okay.
Get your audio and my video.
Okay, so when you bring a specimen, this is the bags that we kept them in. They're just like regular Ziploc gallon bags that you can get at the store. So this label right here is our internal label that we use just to keep our specimens organized. So you can kind of disregard that. But this is what we had it in. And then so if I were going to go to the herbarium with my specimen, I would bring my specimen in the bag. Ideally, well, it would have to be dried out. You don't have to freeze it unless you want to. If you're going to keep it for a while and do one like bulk submission, you can freeze them to make sure there's no insects or parasites on them. But then just keep them in the bag and that'll keep them dry. And then you'll also want to bring your label. So all that you'll need is this paper part. You don't need the background paper or this barcode. And it doesn't need to be written out exactly like this. Whatever information you have is fine. Ideally, just locality. So what county it was in Texas, so state, a little bit of information on where it was found, and then GPS, if you have that, would be the best. And then you put your name down at the bottom because you were the collector and the date that you collected it. And so if you don't have to have this Fungi of Texas on top or this University of Texas herbarium, they will retype this and print it out on this file format. So just bring a general label just so that they have the information. So you'll bring your bag with your specimen and then a label with as much information as you have. Okay. And then George or whoever is working there will put it in either a packet or these Ziploc bags are archival quality, which just means they don't have any coating in it, but it's generally just a Ziploc bag. And so this was the exact fungus that I showed before. You can see it is very small and you can barely make out any features. Like it has a partial veil, but you can't see it in the dried specimen. So that's why it would be good to say it did have a partial veil. You can still see the color, which is good. And it's okay if it does come apart. It will do that. It's dry and it is fragile.
So if they're growing in aggregate, you want to make sure that you get a clump versus a single?
Either would be fine. A clump is just good. So if you forget to write down that it is growing in these clusters, then that would be good to have it already there in a cluster. But no, if you just want to grab one, that's completely fine.
And finding like, sometimes you'll see them with like their various stages of growth too. So that's always
Oh,
good.
yeah. Definitely. Yeah. So if you see one, if you're lucky enough to find like a group of them that are different stages, so one where, it's new and maybe the veil hasn't broken yet, so the gills haven't fully unfolded, one that's mature and one that's maybe a little past maturity, you can collect all of them and bring them all in. So you have different stages represented. Scientists really like that. And plus it's just cool to see. Yeah.
Do we want to take a quick pause? Does anybody have any other questions? And if you don't mind repeating the questions too, since we don't have audio
Yeah.
in the classroom.
Does anybody have any questions?
Will you just touch on briefly the key pointers for photo documentation of the location
Yes.
and the substrate? And then if we have an opportunity to see different stages, things like that. I
Yeah.
know that's covered in another class in more detail.
No, but it's
for
good
the
to touch it
archival
here.
purposes,
Yeah.
I
So
know.
if you have, let me pull up, I actually have some things I can show you regarding that.
And Uncle Don B asked on the chat, how would one preserve a variety that deliquesces? So like an ink cap. for
Oh,
example.
that's a good question. You'd want to collect it early. So if it's already getting inky and smushy, you wouldn't want to collect it. I would want to collect it only young, so at the younger stage, and then dry it immediately. You wouldn't want to wait. It should be fine, but they are usually pretty fragile, so it'll likely kind of start to disintegrate, but that's okay too. As long as you have all the pieces together, it's kind of expected that the more fragile ones won't stay together, and that's okay. Good question. Okay, share my screen. Okay, so I have some photos here. So whenever I collect specimens, I give them a unique identifier. So here, SLP represents, these are all samples that were collected from Stengel. Let me just close this. Lost Pines, which is in Bastrop, that's one of our field stations. And so when I'm out, I usually, you want to get photos of the caps. And here you can see the substrates. You want to be sure you get a photo of the substrate, of the caps. Here I picked them up. Getting a photo of the gills is really important because there are some defining traits that can be determined from the gills. And then also it gives a really nice photo of the annulus, or the veil, the partial veil. Okay, and then you can see between these two photos, you can see the difference in color on the caps versus the gills and the stipe. So you get kind of like a full image of what the mushroom looked like when it was fresh that you do not get when it's dried. And then I took duplicates. I always take duplicates because a lot of times mine come out blurry. Okay, and then just to show you guys how I do this, I have a Excel spreadsheet that I use, in which I put all the information. I have my sample numbers, the species, ignore all the blank spaces, please. I haven't gotten around to identifying them yet. And then I have GPS location and any other information that I have. So data I collected it. This one is whether I collected the specimen. So sometimes I will just note that I did see it. Whether I did a spore print, I don't do it for all of them. Just it depends on time and if I know what it is or not. Whether I took photos, and that's just so later I don't spend forever looking for photos that I don't have. Spore color, if I took the spore print, or if I'm able to discern that from the field. Dispersal strategy, so it has gills or pores or teeth. The substrate.
So maybe we could, I like this format, so maybe
Yeah.
we could share this with everyone.
Oh yeah,
like
that's
a-
good.
Like a spreadsheet, we can put it out on our Google Drive and people can download it if they like the format.
Yeah, that's good.
And I can see like Because I like to use iNaturalist to just log all the GPS data.
Oh, yeah.
So
That's
use
really good.
My key would be like the sample rather than the internal
Yeah,
key that you all go by, like just use the iNaturalist unique ID
that's
number.
really good idea.
And I noticed too,
I don't know if people
what I
online are
was
picking
thinking,
this up.
and maybe we can talk a little bit about this once we get into the Mycoportal stuff, but I noticed that it pulls in data from Mushroom Observer and iNaturalist. And I didn't realize
Oh,
that
yeah.
it did that. And I was like, well, I wonder if I mean, I don't know how, I think it's only research grade things, or I don't know if there's like a way for some, like an admin within Mycoportal to pull in certain observations. But I don't know if it pulls in like all the photos and everything, but I'm just curious because there's a lot of people that like to use that as a tool since it's so user friendly. If there's a way to kind of like integrate that into Mycoportal in
So
a
it
way.
does. So iNaturalist is integrated.
Okay.
And so if you do use iNaturalist, if you do write down your iNaturalist unique identifier that is with your iNaturalist observation, that can be, and when you submit it, you add that to your label. That can be pulled into Mycoportal. So people can then click on the link and go outside to your iNaturalist observation and actually look at it. Yeah, so I can share this. This is just how I do it, so you can modify it to fit your own needs. I mainly collect at the field station, so I do have this organized by location. You don't have to do that. This is just one example. You can make it your own. Okay.
Yeah, did we want to maybe do some practicing labeling?
We could do that.
Since we're not going to start the Mycoportal class
Oh,
until
until
7,
7.
we can give people time that,